Okay, so think about this for a second. When something new comes into being, right, like something completely new, was its form always kind of there, just hidden, waiting to be revealed? Or did it take shape, gradually from something much less defined, much more formless? This
Hmm.
question that's really bugged philosophers for ages.
It has. It really has.
And today we are going to take a deep dive into how this very question has shaped the way we understand biology. You shared some really cool sources on theories of biological development. And our mission, I guess you could say, is to really unpack this whole epigenesis versus preformation debate. It's had a surprising impact on how we view life. So let's get into it.
Yeah. This deep dive is going to be all about this core tension. Epigenesis, the idea that biological form, like how an organism looks, emerges gradually, it's a process, versus preformation. That's the concept that the essential form, like the blueprint, it's already there from the get-go. Maybe it just needs to grow, but it's there. And what's so fascinating to me is how your sources trace this debate all the way back. I mean, we're talking ancient philosophers like Aristotle, all the way to, cutting edge biology research today.
Yeah, it really makes you think about what it means for something to be actually new.
Exactly.
And it's not just a brain teaser for biologists, is it? This epigenesis versus preformation thing. It gets at some really deep questions about reality itself. Like, what's real? Is it the final form, the thing we can see and touch?
So,
Or is it the potential, the underlying structure, the possibility of that form? And then how do we even begin to understand the world? Is it through watching how things develop step by step? Or is there some like preordained order we're supposed to figure? It's pretty deep stuff.
yeah.
And on top of that, the very language we use can be tricky.
Yeah.
Like, take evolution, for example. That word hasn't always meant the same thing. Sometimes it's been used in a preformationist way, other times more epigenetic. So yeah, that adds another layer.
Yeah.
But for this deep dive, we're going to zero in on how individual organisms develop.
Yeah.
From a single cell to a full-fledged being, that kind of thing.
Okay. To even begin to sort this out, this whole philosophical, biological puzzle, it makes sense to start way back. And Aristotle is a great place to start. He's really foundational for the whole epigenesis idea. And it's so cool how he connected his philosophical ideas to actually watching nature.
Yeah, he was a big observer.
He was. So when he looked at, let's say, a chicken developing from an egg, what did he see? A process, stages. So to explain that, he came up with this concept of, get this, four causes. First, you have the material cause, like what's the thing made of, basic.
Right, the ingredients.
Exactly. Then there's the formal cause. That's like the blueprint, the essence of the thing. What makes it that thing?
Okay, so that's like the plant.
Yeah, the plan. Then you've got the efficient cause. What brings the thing about? What actually makes it happen?
So like the builder.
Pretty much. And
Mm.
finally, there's the final cause. This one's about purpose. What's the end goal? What's it supposed to be?
Wow, okay. So he really thought this through.
He did. And for him, observing a chicken embryo, he clearly saw that early on, that egg. It didn't have a mini chicken just hanging out, waiting to get bigger.
Yeah.
The chick's form, its shape, it came about gradually. That was a big moment, a key early statement of epigenesis.
Yeah, that makes sense. So how did Aristotle think this whole development thing got started in the first place?
Well, when it came to reproduction, the birds and the bees,
Right, right.
Aristotle saw it as a team effort, a combination of contributions from both male and female.
Okay.
And this combo, this union, that's what kicks off the whole developmental process, the journey.
Okay, so what did each side bring to the table?
So he considered the female contribution to be the material, like the basic building blocks, the raw ingredients, if you will. And he associated that with menstrual blood.
Okay.
But it was the male's contribution, the semen, that provided what he called the principle of generation.
Okay, I like that. The principle of generation.
Right. It's the active force that shapes the material. So the female provides the stuff, and the male provides the instructions, the guiding force.
Wow, that is definitely different from how we think about it today.
Oh, it totally is. But remember, we're talking about a completely different time and a totally different way of looking at the world. And to connect this to his broader philosophy, Aristotle also thought about the soul. He saw it as this inherent source of motion inside every organism, like driving this gradual epigenetic development.
Okay, so when he says soul, he doesn't necessarily mean like, our modern religious idea of a soul, right?
Exactly. It's not about, heaven and hell. For Aristotle, the soul was more like the set of essential functions of a living thing. And it had different layers, depending on the organism. So plants have what he called a vegetative soul for basic life stuff.
Right.
Animals that move around have a locomotory soul on top of that. And then us humans, we've got the rational soul, which is all about thinking and reasoning. But the point is, he believed this inherent soul, whatever its flavor, was present right from the start, in that combined male and female contribution. And it was the driving force behind how form and function unfolded over time. So for him, the potential for the fully formed organism was there, but it had to be activated. It had to be realized through this internal guide, this soul.
That's a really interesting way to think about it. And what's amazing is how these ideas from Aristotle, they didn't just disappear, like some ancient philosophy textbook.
Yeah.
They had a huge, lasting impact on Western thought.
Mhm.
Even traditional Catholicism for centuries, like figures like Augustine and Aquinas, they were heavily influenced by Aristotle. And they believed that human development, what they called hominization, was also this gradual thing. They even had this stage called quickening
Yeah.
around 40 days into development. And that was when this material, which they initially thought of as more animalistic, was believed to be infused with a soul, a human soul. It really makes you think, Because it wasn't until 1859, relatively recently, that the church officially shifted away from this earlier epigenetic view
That's right.
and declared that life begins at conception. So that's a huge example of how these abstract biological ideas have had really significant consequences,
Yeah.
cultural, religious, even political.
It's incredible. Okay, now let's do a time jump. Fast forward a bunch of centuries to the 18th century. So the intellectual scene has changed a lot, right? There's this big fascination with natural history and this new tool, the microscope. It's like a whole new world opening up.
Yeah, being able to see things we never could before.
Totally. So as the 18th century kicks off, that Aristotelian epigenesis, still a big deal. Researchers are all about observing this gradual emergence of form from what seems like, pretty undefined material. But here's the thing, like we touched on with Aristotle, this epigenetic view, it often needed this idea of a vital force.
Right.
This non-material thing that explains how unformed matter could, spontaneously become a complex living being.
I see.
It's like, where does that organization come from? But now this idea of a vital force, it's bumping up against the new kid on the block materialism.
Right, the idea that everything's just matter and energy.
Exactly. So if you're thinking everything in the universe is just physical stuff in motion, then how do you explain something as intricate and organized as a living organism coming from something seemingly simple?
Yeah, that's a tough one.
It is. It's a big challenge for folks who want a purely material explanation of the world.
So what happened?
Enter preformationism. It makes a comeback, and it comes back strong. It's seen as a pretty compelling alternative. And there's this image that really sticks with people, this idea of the homunculus.
Oh, yeah, I've heard of that.
So imagine a tiny, perfectly formed little person. Some folks, the spermists, they thought this homunculus was tucked away inside the sperm.
Okay.
Guys like Hartsoker, they were drawing these detailed pictures of it, and then you had the ovists who thought the miniature person was hiding in the egg.
So the debate was about where the tiny person was, not if it was there.
Pretty much. And this was a literal take on preformation. The adult form was physically there from the beginning, just tiny. And development was just this little guy, this little gal growing bigger. It's kind of a mind-blowing image when you think about it, right?
It is. It really is. But to be fair, not every preformationist was like picturing a tiny human in there.
Oh, definitely not. It wasn't always about that literal image.
Right.
The core idea was this. The organization, the blueprint, it was pre-existing in some form. And some thinkers argued that this idea actually fit quite well with a materialist worldview. Everything's just matter.
Right.
Because if the form was already there, then development was more like a mechanical process, like a clock unwinding. No need for that mysterious vital force to keep making new stuff.
Okay.
And this had some appeal for religious folks, too. They saw it as evidence of, a preordained divine plan. God put the little person in there.
That's interesting.
It is. And this back and forth really comes into focus when you look at two guys, Caspar Friedrich Wolff and Charles Bonnet. Wolff was all about epigenesis. He was meticulously observing chick embryos, watching form emerge step by step. But then you have Bonnet, a devoted preformationist. He's seeing those same stages of development, but he's convinced the form was always there, just unfolding, getting bigger.
So they're looking at similar things, but they come to opposite conclusions.
Exactly. And it shows how our big picture ideas about reality and how we learn about it can really shape our scientific views. Wolff is all about direct observation, seeing the form emerge. Bonnet is relying on this belief that the form is pre-existing. It's like two different lenses on the same world.
Right, like two different filters.
Totally. Okay, now let's roll into the 19th century. Things get shaken up again, and guess what plays a big role?
I'm guessing evolution.
You got it. Darwin's work in 1859, his ideas put embryos in the spotlight. They became key to understanding how different species are related, the family tree of life.
Right.
And then there was this guy, Ernst Haeckel, who was a real showman.
Oh, yeah, I've seen his drawings. Those intricate, beautiful embryo illustrations.
Those are the ones. So Haeckel was a huge popularizer of embryology. He connected it to evolution big time. He pushed this idea that ontogeny, which is the development of a single organism, basically replays phylogeny, which is its evolutionary history. So as you develop, you're going through stages that look like the adult forms of your ancestors.
Wow.
It's a pretty wild idea, right? And this strongly implied a kind of pre-scripted development, like a pre-recorded movie playing out, definitely leaning towards pre-formationism. Part of this was Haeckel's own thing. He was a monistic materialist. He believed everything was ultimately one substance, and form emerged by following this predetermined evolutionary path. It was like it was encoded right into you.
Okay, so for Haeckel, evolution wasn't just about how species changed over time. It was also built into the very way individuals developed.
Right. Now, to be fair to Darwin, while he definitely used those similarities in embryos as evidence for evolution, he didn't really take a hard stance on the details of individual development, whether it was strictly epigenesis or preformation. He saw the connections, but he didn't dive into the nitty-gritty of how it all worked.
Yeah, he was more focused on the big picture of evolution itself.
He was. And it was Haeckel who really ran with this more preformationist interpretation within that evolutionary context. But his views, while popular at the time, weren't necessarily based on a ton of new observations of embryos. They were more driven by his own philosophical ideas, and he wanted to provide this really strong visual evidence for evolution, which, to be fair, he did.
Right, those drawings were very convincing. Okay, so we're set up for even more debate then.
Oh, yeah. And things get really heated in the late 19th century. And that's where we get figures like August Weismann and Oscar Hertwig. And it's fascinating to see how, even within science, you have these strong opposing viewpoints on something so fundamental.
Yeah, like what determines how an organism develops.
Exactly. And in 1899, this guy, William Morton Wheeler, he makes this really insightful observation. He basically says there are two basic types of thinkers in biology. Those who are all about change, process, interaction. They're more into epigenesis, physiology, how things work. And then you have those who are focused on stability, pre-existing form, structure. They lean towards preformationism, morphology, what things look like.
Interesting. So it's like two different mindsets almost.
It is. And he argues these tendencies, they stick around, they shape how we try to understand the living world, even as our specific biological theories change. Wheeler himself felt that neither extreme was enough to explain development. He called for a more integrated view, one that took both predetermination and emergent development into account. And he stressed that we needed real scientific evidence to figure out how much each mattered.
And Weismann himself is a great example of this tension. He actually started out leaning towards epigenesis. He wasn't buying the idea that the complex form of an adult could just unfold from pre-existing stuff in the germ cells. But then, as he dug deeper into cell theory and inheritance, his thinking changed big time.
It did. And it's so interesting to read his own account of this change. At first, he thought the idea of preformed parts for every single bit of the body, all crammed into those tiny germ cells, was way too simplistic, even logically impossible. He wanted a way for that complex adult organization to emerge gradually, epigenetically, from something simpler in the germplasm. But he kept trying to make that work with what he knew about biology, and he just couldn't. He became more and more convinced that epigenesis alone couldn't explain how development was so precise and predictable.
So it's like his own research pushed him in a different direction.
It did, and this led him to his famous germplasm theory.
Okay, so what's that all about?
Well, he proposed that the germ cells, those special cells that become eggs and sperm, contained this unique substance, this germplasm, and this stuff held all the instructions, all the determinants to direct development. He saw inheritance as the key, the driver of differentiation. These determinants inside the germ cells were calling the shots, predetermining what each part of the developing organism would become. He even compared cell division to a mosaic.
Okay, I like that.
Right. Each daughter cell, when a cell divides, it gets a specific set of these determinants, like tiles in a mosaic. And that determines its fate, its role in the developing embryo.
So basically everything's pre-programmed in the germplasm.
Pretty much. Weismann argued that what a cell becomes isn't decided by its surroundings or its neighbors. It's all about those internal factors, those inherited determinants from the germplasm. This was a strong statement for preformationism.
Yeah.
The blueprint for development was basically set in the germ cells right from the start.
So it's like inheriting a detailed instruction manual.
Right. But now, not everyone was on board with this. Oscar Hertwig, for example, he was a vocal critic of Weismann's ideas.
He didn't buy it.
He did not. He argued that Weismann wasn't really explaining anything. He was just pushing the problem somewhere else into this invisible region. The germplasm and its determinants, which at the time you couldn't even see or study directly. It was all theoretical. Hertwig was all about studying what you could observe. He focused on those dynamic interactions between cells and how differences arise through those interactions as the real source of differentiation. For him, complexity wasn't pre-built in the germplasm. It emerged over time through these dynamic, interactive processes happening between cells. He even compared cells to little interacting organisms that build the whole thing together.
So it's like a community effort, not a solo act.
Exactly. So while Weismann was picturing development as this self-differentiation guided by internal instructions, Hertwig saw it as way more interactive and emergent, shaped by both what's inside that fertilized egg and the environment around the cells. And this highlights that fundamental philosophical difference we keep seeing. How do you think about the very nature of a developing organism? And like Wheeler said, it seemed to come down to different basic assumptions about how biological complexity happens. And we needed new experiments to shed more light on the question.
Okay, so new experiments. Who's stepping up?
Well, this is where we get Wilhelm Roux and Hans Driesch. They were doing this groundbreaking work in the late 19th century, and at first, Roux's experiments on frog embryos seemed to support a more preformationist view, kind of like Weismann's mosaic development idea.
Okay.
He did this famous experiment where he damaged one of the first 2 cells of a frog embryo, and the remaining cell, it seemed to develop into only half an embryo.
So he's preventing one cell from developing, and he gets half a frog.
Basically, yeah. He stopped one of those first 2 cells from developing further, and what he got looked like half a larva. And this led him to think that each of these early cells already had a set fate, like those tiles in a mosaic, each destined to make a specific part of the whole organism.
Okay, so he's seeing evidence for pre-programming.
He is, but then comes Driesch. He's working with sea urchin embryos, and he does something different. Instead of just damaging one cell, he completely separates those first 2 cells of a fertilized egg.
Okay.
And guess what? He doesn't get 2 half embryos.
He gets...
2 smaller but complete larvae.
Wow.
Right. It was a huge deal. This directly challenged Roux's mosaic idea. It suggested that those early cells were totipotent,
Right.
meaning each one had the potential to become a whole organism.
Oh, wow.
Yeah. This pointed to a much more flexible, epigenetic kind of development. The fate of those early cells wasn't fixed. It depended on their interactions, their position, how they talked to each other. And at first, Driesch took this as supporting a materialistic, epigenetic view. Those early cells could make a whole organism, and their interactions during development led to differentiation, to becoming different cell types. But it's interesting, he actually shifted towards a more vitalistic view later on, like he thought there was some non-material force guiding it.
So even he was grappling with those big questions about what drives development.
He was. And you would think Roux would have to admit he was wrong after Driesch's experiment. But he didn't fully abandon his mosaic idea. Instead, he added this idea of reserve idioplasm.
Okay, what's that?
It's like saying, even in a mosaic system where cells have a preset fate, they might have this backup genetic material just in case. It's like a safety net, allowing for regeneration or adaptation if needed.
So he's trying to explain Driesch's results without completely giving up on his original idea.
He is. But as more people weighed in, like Wheeler and Whitman, they started pushing for a middle ground.
Yeah.
Recognizing that there's probably some internal programming, some predisposition, but also that organisms can respond and adapt to their surroundings during development.
So it's not either, it's both.
Right. And this leads to a big shift in the early 20th century. It's not just preformation versus epigenesis anymore. It becomes about regulative theories of development.
So what does that mean?
Basically, the question becomes, how is development controlled? Is it mostly the genes calling the shots, predetermined path? Or is it more about self-organization, like cells interacting and figuring things out as they go?
So it's getting more nuanced.
Much more. And if genes are seen as the boss, then development has this preformationist feel to it. It's like the genes have the blueprint and they're just carrying it out. But if it's more about self-organization, that's more epigenetic. Like the environment and interactions matter a lot.
I see. So the debate is evolving.
It is, and the first half of the 20th century sees the rise of these regulative theories. One big one is organicism.
Okay, let's talk about organicism. What's the gist?
It's a shift in how biologists are thinking. Donna Haraway, a historian of biology, she called it a new paradigm for embryology.
Okay.
So organicism says the organism is this whole interconnected thing, right? Parts working together at different levels. And development is goal-directed, but it's not necessarily fixed by genes. It's like there is a goal, but there are multiple ways to get there.
I see.
And instead of just looking at genes as instructions, organicists wanted to understand form through biological principles and relationships.
So it's a more holistic view.
Very much so. And you have thinkers like Immanuel Kant who talked about this idea of reciprocal determination in living systems, like everything influences everything else. Then there's Ludwig von Bertalanffy, who developed general systems theory, thinking about systems as a whole. And Darcy Thompson, he wrote On Growth and Form, which was super influential. He looked at how physical forces shape form. The big idea here is that form emerges from a self-organizing system, not just genes dictating everything.
So it's a lot more dynamic than just following a blueprint.
Absolutely. And within this organicist way of thinking, ideas like gradients and fields become really important for explaining how complex shapes and organization arise during development.
Okay, tell me more about that.
So organicists use these more abstract concepts like symmetry, polarity, gradients, and fields to explain how form emerges. For example, Hans Spemann, building on those organizer experiments, he thought about morphogenetic gradients along the axis of the embryo. It's like imagine a gradient of a chemical, and it influences what cells become along that line. And Charles Manning-Child, he had this idea of activity gradients based on how active the metabolism is in different parts of the developing organism. He thought that played a big role in driving development. But these concepts weren't always accepted. Some people thought they were too vague, they didn't have a clear physical basis, or they were worried it was a backslide into vitalism, that non-material force idea.
Right, like maybe they were sneaking that back in.
Exactly. But the ideas about gradients and fields did get some solid experimental support. Remember Hild Mangold's organizer experiments with newts?
Yeah, she was the one who transplanted a bit of tissue and got a whole new body axis to form.
Exactly. That was a big deal. That tiny piece of tissue, the organizer, as Spemann called it, could direct the development of a whole new structure. And the way they thought it worked was through a gradient of some chemical, still unknown at the time, that organized the cells around it. Right. This really boosted the idea of field theories in embryology, the idea that the early embryo is organized into these dynamic fields, kind of like invisible landscapes with peaks and valleys of influence. And the position of a cell within that field, its distance from the organizer, determines what it becomes.
Okay, that's a cool way to think about it. So what about Joseph Needham? He was a big name in the organicist movement, wasn't he?
He was. And he was really trying to figure out the chemical basis for this organizer effect and to make the concept of developmental fields more rigorous. He was a biochemist, so he was thinking about the molecules involved. He drew on Darcy Thompson's work on physical forces and form, and he came up with this idea. Developmental fields are like regions in the embryo that determine what kind of development happens there, its direction, its intensity. He imagined development as moving through these stable states within these fields, governed by what he called field laws.
So like rules of the game, almost.
Right, but he didn't really nail down what those laws were exactly. Still, his ideas were important. They foreshadowed those systems-level approaches that we use today to understand development.
Okay, so the picture's getting more complex.
It is. And then there's Paul Weiss in the U.S. He also contributed a lot to this gradient and field thinking with his theory of molecular ecology.
Molecular ecology, that sounds interesting.
It was very ahead of its time. He said that inside a cell, there's this whole mix of different molecules, and their arrangement is determined by how they interact with each other and with the physical environment around them.
Okay.
But he didn't think it was just random. He thought these interactions were organized by higher-level developmental fields. This led to those predictable patterns of cells becoming different, growing and forming structures. He called it gradual determination. It wasn't like one spot was controlling everything. The organization emerged dynamically through these gradients and interactions.
So again, it's this idea of emerging complexity, not a preset plan.
Right. And his work on limb regeneration in amphibians was really influential for this. He showed how those regenerating tissues could get the right orientation, even if they came from different cell types and locations. It's like they knew where they were supposed to be and what they were supposed to do.
Pretty amazing. Okay, so we're seeing a lot of focus on these fields and gradients. But then in the 20th century, we also get some really different approaches. Like Alan Turing, the computer science guy, he comes up with this reaction diffusion model. That's a very mathematical way to think about how patterns could arise, very different from the more biologically focused organicist views.
Oh, absolutely. Turing was brilliant, and he tackled this big question. How do these complex patterns appear in an embryo that starts out looking pretty uniform?
Yeah, good question.
So he came up with this reaction diffusion model. It's based on equations that describe how two hypothetical chemicals, which he called morphogens, interact and spread through a tissue. One acts as an activator, the other as an inhibitor. And the key thing is they have to diffuse at different rates. And what he showed is that stable spatial patterns, like stripes or spots, could emerge from a tissue that started out homogeneous, all the same.
That's wild. You'd think diffusion would make things more uniform, not create patterns.
Exactly. That's what's so cool about it. He showed that diffusion, when combined with these local reactions and this kind of lateral inhibition, could create structure. It was a huge insight. Now, his model was very abstract. He didn't say which actual molecules in the embryo were these morphogens or exactly how these chemical patterns led to cells behaving differently, but it had a huge impact. It spurred a ton of research into reaction diffusion mechanisms in all sorts of developmental phenomena, from the patterns on animal coats to how things like hair follicles and fingers form.
So it's like he provided a theoretical framework that people could then test in real biological systems.
Exactly. But the model did have its critics. One issue was this assumption that the starting tissue was perfectly homogeneous, which isn't always the case in real embryos.
All right, it's a simplification.
It is. And then there was Francis Crick, one of the guys who discovered the structure of DNA. He also took a crack at explaining pattern formation with his source sink model.
Okay, so another take on diffusion.
Right. His model was simpler. It focused on one morphogen diffusing from a source to a sink along a line of cells. And the idea was that the concentration gradient of this morphogen would tell the cells along that line what to become. So it's like a signal that gets weaker as you move further from the source.
Okay, I see.
It was elegant, but some people argued that it wasn't powerful enough to explain those really complex patterns you see in real development.
So too simple.
Maybe. Now, another big concept from the mid-20th century was Louis Wolpert's idea of positional information. This was a really important attempt to bring together the gene-centric view and those spatial ideas from the organicists.
Okay, so a bridge between the two camps.
Yeah, exactly. Wolpert said that each cell in a developing field, it figures out its identity based on where it is in a gradient of a signaling molecule. So it's like the cell gets a signal that says, you're here, so become this. He called this positional information.
Makes sense.
And he saw it as a fundamental mechanism for translating genetic information into spatial patterns. So the genes provide the instructions, but this positional information tells the cells how to arrange themselves in space.
Okay, so it combines genes with spatial organization.
It does. And then cells would interpret this positional information by turning on or off specific genes, leading to differentiation and morphogenesis. So it's a really neat way to link genes to the actual shaping of the organism. But Wolpert's thinking on this, it changed over time.
How so?
By the 1970s, he was putting more and more emphasis on genes. He started talking about a genetic program for development, and he saw the embryo as this complex system that could be predicted from the instructions in the genes.
Okay, so back to the blueprint idea.
Kind of, but now the blueprint was in the genes. This led to this whole field of computational embryology, where people were trying to figure out how to, compute an organism's form from its genes.
Oh, that's ambitious.
It was. And this reductionist view, it had its supporters, but it also had its critics. Some argued that it was basically preformationism in a new disguise. The form was still predetermined just by the genes now. And they pointed out that it still didn't explain how the actual shaping happened, the morphogenesis part, or how the environment could influence things.
So it's like they were saying, okay, the genes might be the blueprint, but where's the builder? And what about the building materials?
Exactly. But you can't deny the impact that genetics had in the 20th century. It really brought in a new kind of predeterminism, this focus on those genes inside the nucleus as the main drivers of development.
So it's like genes took center stage.
They did. And while early 20th century embryology was leaning towards those epigenetic mechanisms and cellular interactions, genetics shifted the focus to the nucleus as the control center. But unlike those earlier preformationist ideas, like Weissman's mosaic thing, this new genetics said that every cell in an organism has the same set of genes. So the question became, how do you get all these different cell types if they all have the same instructions? The answer they came up with was that different genes are turned on or off in different cells based on some internal program. This gene-centric veer was appealing because it focused on these tangible units of heredity, the genes themselves. You could study them, manipulate them.
This is a more concrete way to think about inheritance.
It is.
Yeah.
And even Thomas Hunt Morgan, who was a giant in early genetics, he was hesitant about this simple Mendelian chromosome theory at first. He came from embryology, and he just couldn't believe those identical chromosomes in every cell could explain all the complexity of development. He kept saying that both genes and those dynamic changes during development were important.
So he was trying to keep